नमस्कार, आप सभी का एक बार पुनः स्वागत है। मैं हूं राहुल और आप देख रहे हैं पीएम eVidya चैनल्स और एनसीईआरटी का ऑफिशियल यूट्यूब चैनल। यू आर वाचिंग अस ऑन चैनल नंबर 12 एंड today we have a live interactive session of biology and the topic of discussion is chapter number 2, sexual reproduction in flowering plants. इसके बारे में हमने पहले कुछ और सेशंस किए हैं जिनमें पढ़ा है, लेकिन आज और हम इसे जानने की कोशिश करेंगे कि आज कैसे हम इन फ्लावरिंग प्लांट्स के बारे में जानेंगे, इनका जो सेक्सुअल रिप्रोडक्शन है। क्लास 12 का ये लाइव इंटरेक्टिव सेशन है। आप भी चाहें तो इसका हिस्सा बन सकते हैं। ईमेल आईडी और फ़ोन नंबर का प्रयोग करेंगे आप। हमारा फ़ोन नंबर है 8800440559 और क्लास 12 का ईमेल आईडी भी आप उसे ज़रूर अपने पास रखें। वी हैव डॉक्टर पूजा गोखले सिन्हा, शी इज़ असिस्टेंट प्रोफेसर in Department of Botany, Sri Venkateswara College, University of Delhi. Dr. Pooja is in the studio with us. Dr. Pooja, namaskar and a very warm welcome.
Thank you, Rahul.
Good to have you here again. पहले भी हमने एक सेशन किया है. I'm sure वो भी क्लास 12 का ही था. I'm sure या 11th का था. 11th का था. बच्चों ने उसको enjoy इसलिए किया था क्योंकि वो समझ रहे थे. कोई भी सब्जेक्ट तब आसान हो जाता है जब वो समझने लगते हैं. If there is no burden, तो आज कैसे ये फ्लावरिंग प्लांट्स के बारे में कैसे हम पढ़ेंगे ताकि क्लास इंटरेक्टिव जो है वो इंटरेस्टिंग भी रहे बच्चों के लिए।
फ्लावरिंग प्लांट्स के बारे में वी आर गोइंग टू स्टडी अबाउट देयर सेक्सुअल रिप्रोडक्शन टुडे। और हमने क्लास 11th में ये बच्चे ऑलरेडी पढ़ के आए हैं व्हाट आर द डिफरेंट पार्ट्स ऑफ अ फ्लावर, हाउ उसको डिफाइन कैसे करते हैं, व्हाट आर द टर्म्स दैट वी यूज, एंड दे इन दोज सेशंस आल्सो आई टोल्ड देम टू ऑब्जर्व प्लांट्स अराउंड देम एंड देन डिफाइन देम, डिस्क्राइब देम. Now we are in this unit we are going to study the functional aspects of these flowers. What actually flowers do for plants, especially the flowering plants which we call angiosperms. So chapter number 2 of NCERT class 12 biology, it talks about the processes which are involved in sexual reproduction of flowering plants. So this is about the unit that we are going to discuss today, and it is a very relevant unit, Rahul. It is very important from children's perspective, and it is also very interesting. And again, plants are something which they are everywhere around us, so students need to be a bit more observant and just observe things around them, so they can very easily relate to what functions and processes we will be talking in today's session.
Okay. So let's explore how we are going to do this.
Yeah, let us start with the presentation now.
Please.
So now coming to the presentation, the current topic, the topic that we are going to talk about today is sexual reproduction in flowering plants. It is chapter number 2 of the NCERT book. In order to just introduce our students to what actually flowers are, we have already done in class 11th the basic outline of what are the different floral appendages, the floral whorls, and what are the different parts of a flower called. Now what they actually do for a plant is to conduct sexual reproduction. Now sexual reproduction is very important for survival of species, and when plants reproduce sexually, so the next generation, the progeny, it is better than the previous generation because of several reasons. Because there is genetic mixing of different parents, so the progeny that is formed, it is more robust and it is better than the parental generations. Sexual reproduction, it brings variability, it gives adaptability for plants to survive, and also it makes them more competent and more vigorous and adaptable to survive in different conditions. Now flowers are something which we all have seen. None of us is there who has not seen a flower.
Right.
They are the organs which are actually responsible for sexual reproduction. They occur in different forms, shape, size, colors, and there is a lot of variation. If we just look around ourselves, then we see a large number of flowers which are very variable in the way they look. The fragrance is different, the color, and they look totally different from each other. But irrespective of how they look and what is their size, color and shape, fragrance, all the flowers in plants, they conduct the process, they are used in the process of sexual reproduction in plants, which is very, very important.
Okay.
Their main function is to produce seeds and fruits, because the seeds which are produced inside the fruits, they are responsible for giving the next generation of that particular plant. Right. Seeds germinate, and those germinated seeds, they give new plantlets, and those plantlets then grow up to form new adult plants. Right. So what a flower does is actually harbor seeds and fruits, and then that is responsible for the next generation.
Okay.
Now coming again to the basic structure of a flower, all the students, they have been doing this structure since class 6th, but again just to revise, let us just look at this flower. It is a longitudinal section of a flower, and the flower, you can see it has the different whorls. The sepals are there, which are the green colored structure which cover the bud. Then there are petals, which are the colored structures which we see when the flower open. Then in the center there is the carpel or the pistil, which has style, stigma and ovary. Now this particular region is the female reproductive part of a flower. And the stamens, which are arranged around the carpel, they have anther and filament. So these represent the male reproductive structure of a typical flower. Flowers have a lot of aesthetic, ornamental, social, religious and cultural importance. So they do a lot for human, but their primary function for the species is the reproduction, and that is sexual reproduction. Now when we talk about sexual reproduction in plants, we can broadly categorize it into 3 phases.
Okay.
The first is the pre-fertilization phase. This phase is a phase in which preparation for the formation of zygote takes place. The male and the female reproductive organ, they produce the male and the female gametes. So formation of the gametes, maturation of the gametes takes place as of pre-fertilization events. Microsporogenesis and megasporogenesis are the 2 events which take place for the preparation of the male and the female gametes respectively. जो माइक्रोस्पोरोजेनेसिस होता है उससे मेल गैमीट फॉर्म होता है, pollen grains फॉर्म होते हैं, and this process it takes place in the anther, that is the male reproductive structure of a flower. And the megasporogenesis, it is responsible for the formation of the embryo sac which has the egg. So जो egg है, which is going to be the female, which is the female gamete of a flower, that formation, उसका development and formation, उसको megasporogenesis कहते हैं. And both these events, they take place in a flower. If a flower has both the sex organs in the same flower, then both these take place simultaneously. Sometimes it happens that a plant has only male flower and sometimes it has only female flowers. So in that situation, either microsporogenesis or megasporogenesis takes place in the flower. The next major step is the step of fertilization. Fertilization is the key step in which the gametes, that means the pollen grain and the egg, which is formed, now fuse, and they fuse to form zygote. Zygote and also the primary endosperm nucleus. Now zygote is the first cell of the next generation, we can say that. And this zygote, during the process of fertilization, when it is formed, the zygote, it again restores the diploid ploidy of the plant. I will like to emphasize here, जब pre-fertilization events होते हैं, microsporogenesis and megasporogenesis, those 2 events, they are majorly, they happen by the process of meiosis. Students should recall, meiosis is reductional division. So जो microspore mother cell या megaspore mother cell होता है, वो diploid होता है. That means all the chromosomes are present in double, in 2 numbers. Then meiosis happens. फिर जो zygote, फिर जो egg and pollen grain बनता है, that is haploid. During fertilization, these 2 haploid cells, they again fuse and they lead to the formation of a zygote, which is diploid. Now, the primary endosperm nucleus in angiosperms, the flowering plants, is very different from other group of plants. This is a triploid nucleus. It has 3 number of chromosomes. All the chromosomes are present in 3 numbers, triplicates. This formation is a very interesting formation and it is exclusively present in the flowering plants or angiosperms. So जो fertilization event है angiosperms में, उसमें 2 fertilization होता है. एक pollen grain and the egg cell fuses to form the zygote, और दूसरा primary endosperm nucleus भी fertilization से ही बनता है. तो fertilization in angiosperms or flowering plants is also known as double fertilization. Students should remember this term very clearly because it is a characteristic feature of flowering plants. After the formation of zygote and the primary endosperm nucleus, now the zygote has to grow into a new embryo which is present in a seed. So, the zygote also changes and also concomitantly the ovary also changes. So, formation of fruit, it takes place, and inside the embryogenesis is taking place. Also endosperm develops. Endosperm basic function is to provide nutrition to the growing embryo, because the embryo is the structure which is going to give rise to the next generation plant. Embryo is embedded inside a seed. So, post fertilization events prepare the embryo to be robust and nutritively competent to give rise to the next generation plantlet. So, this differentiation or this demarcation, when students should, whenever they sit and read this chapter, they should ensure that they demarcate all these three processes. उससे दो फायदे होंगे, एक क्लैरिटी बच्चों को बेटर होगी, एंड ऑल्सो हर एक प्रोसेस के सब स्टेप्स अगर वो अच्छे से लिख ले एंड डायग्राम्स बना ले साथ में, तो इट विल हेल्प देम अ लॉट. स्टूडेंट्स, इन दिस यूनिट आई वुड लाइक टू मेंशन दैट बहुत डिस्क्रिप्टिव यूनिट है ये. इसके अंदर प्रोसेसेस बहुत ज़्यादा हैं और उनको लिखना पड़ता है. अगर वो डायग्राम के साथ साथ लिखें, एंड अगर आप जब अटेम्प्ट कर रहे हैं एंड यू ड्रॉ अ गुड डायग्राम, well labeled diagram, it will not only help them to remember, but also it will help them to score. So this chapter is also can be very scoring if the descriptive part is also supported by good diagrams. And NCERT book has very good clear diagrams, so you should use them to understand, remember, and also write your answers to prepare for your exams.
You have to practice, to draw the the beautiful diagrams for the
for
the examiners. क्योंकि ऐसा कहा जाता है कि जब आप सुंदर बना देते हैं तो वो अट्रैक्ट जो उन्हें करती है, शायद उसमें मार्क्स ज्यादा मिलते हैं.
अगर आपने अच्छा डायग्राम बनाया है तो एग्जामिनर लुकिंग एट द डायग्राम विल अंडरस्टैंड ओके द चाइल्ड नोज द कॉन्सेप्ट. बच्चे को कॉन्सेप्ट आता है तो, और जब आपको खुद को भी पढ़ने में इट विल बी वेरी हेल्पफुल. व्हेन यू ड्रॉ and when you read together, you will remember it for longer time. Only words you may jumble up and forget. So that is the catch in this chapter. Now moving on to the next slide.
Please.
Now, when flowering happens, जब हमें flowers दिखते हैं, so the plant prepares for flowering very well in advance. When we see flowers, that is just a manifestation of several processes which are happening inside the plant. So the shoot apical meristem, जिसके अंदर पहले leaves होते हैं, that decides okay now flowering has to take place. And how it decide is by the stage of the plant की age, environmental conditions, temperature, rainfall. When all these conducive conditions are there, then the plant it gets into flowering. That is why we have flowering of plants in specific season. So season is nothing but a particular range of temperature and rainfall that plants need to flower. So the decision to get into flowering, it is taken place much earlier before we actually see flowers. And the changes they start in the shoot apical meristem. So the shoot meristem, tip of the shoot, that is the region where the initiation of flowering takes place. This initiation of flowering in the initial stages, it is because of changes in hormones, and these hormones they then bring about structural changes. Nowadays we also know about all the genes which are responsible for flowering. So it is a very intricate process. What we see is just a flower, but the plant has to prepare a lot inside it. Then after the plant has decided, the starting of flowering will take place and the structural changes they begin. The first thing that happens is differentiation of androecium, gynoecium along with the sepals and petals, because androecium and gynoecium are the major structures which are going to give rise to the male and the female gametes in a plant. The stamen is the chief organ where the male gamete is produced. It has microsporangium, which is a structure जिसके अंदर male gamete और pollen grain होता है. The male gamete in a plant or the male gametophyte in a plant is known as a pollen grain. So stamen जो है, it is a single unit of androecium. You can see on your screens the tip of the stamen is a lobed structure. वो जो lobed structure है उसको anther कहते हैं. And on the right you can see that अगर हम उसका cross section cut करें तो उस lobed structure को 4 lobes होते हैं उसके अंदर. इन चारों lobes को sporangia कहते हैं और इनके अंदर pollen grain develop होता है.
Okay.
In this unit, not only what is inside the structure, we will also study कि pollen grains बनते कैसे हैं. And the stamens are characteristic for species. Each species will have a particular design, color, length and number of stamens.
Okay.
Now coming to the anther, जो anther है, जो tip है stamen का, a typical angiosperm anther bilobed होता है, 2 lobes जैसे दिखता है वो, और हर उसके अंदर थीकिया होता है, इसीलिए angiosperm anther को diathecus कहते हैं. You have to remember these terms, students. There are a lot of terms in this chapter. If you break this term into their meanings, it will be very easy for you to remember. And अगर साथ में again I'll repeat, if you know the diagram, so you don't even have to mug it up. You will simply understand. It is a structure.
Easily they can, give us some 2 number or 1 number of questions. Ki
yes,
what is
a lot of short notes, a lot of define, lot of exemplaries, they come from this unit. So this unit is, if you understand it well, it is very scoring for students.
और ये भी देखने में आ रहा है क्योंकि काफी टाइम से जैसा आपने कहा कि बच्चों को कंसेप्ट आता है, examiner can examine easily.
Yes.
Just watching or checking the
neatness of your answer and the diagrams.
तो इस तरीके से मुझे लग रहा है वो IQ चेक करने के लिए भी ऐसे क्वेश्चंस फ्रेम कर सकते हैं जो कि ट्रिकी होंगे एंड आई एम श्योर बहुत आसान होंगे, लेकिन यदि आपका कंसेप्ट क्लियर है तो ही आप इसे कर पाएंगे.
यस, आप उस वर्ड को, उस टर्म को डिवाइड कर लीजिए, उसका मतलब समझिए, देन यू विल डोंट, आपको रटने की जरूरत नहीं पड़ेगी, यू अंडरस्टैंड इट. एंड देन फ्लावर द ब्यूटी अगेन, आप आसपास जाके देखिए, आपको ये सब दिखेगा. इट इज एवरीवेयर अराउंड अस. नाउ कमिंग टू द एंथर टाइप. अगर हम ट्रांसफर सेक्शन काटते हैं एंथर का, एंथर टिप का, तो हमें दिखता है कि वो 4 लोब्स जैसे होते हैं, जैसे हमने पहली स्लाइड में देखा था. तो 4 साइड्स जो होते हैं उसको स्पोरेंजिया कहते हैं, क्योंकि उसके अंदर माइक्रोस्पोर्स होते हैं. अगेन स्पोरेंजिया, इट इज अ स्ट्रक्चर जिसके अंदर स्पोर्स होते हैं. माइक्रोस्पोरेंजिया जिसके अंदर माइक्रोस्पोर्स होते हैं. Micro is pollen से related. So this is how you remember things very easily. So each lobe, it has 2 microsporangia, and these microsporangia, when they mature, so they have the pollen grains. Look at this structure on your screen. This structure is a section of a young anther, और आपको दिख रहा है कि wall है जो कि layered है. एंथर की जो वॉल होती है, इट इज़ अ वेरी इंटरेस्टिंग स्ट्रक्चर एंड लेयर्ड होती है, बिकॉज इट हैज़ टू प्रोटेक्ट द pollen grains, because if pollen grains are not protected, then the process of fertilization will not take place. So when the anther is mature, it has 4 developed pollen anther wall layers. These 4 wall layers, if I start from the outside, so सबसे बाहर वाली लेयर होती है एपिडर्मिस. Then there is the endothecium. After the endothecium, there are 2 to 3 layered cells, and this 2 to 3 layered structure is known, they are known as the middle layers. And the innermost layer of the anther wall, it is known as the tapetum. Now all these 4 structures, they have very peculiar functions. The first 3, epidermis protects the anther. Endothecium and middle layer, they help in the dehiscence, because when anthers dehisce or they open up to release the pollen grain. So these two layers, they help in the process of dehiscence or breaking up of anther. Coming to the innermost layer, the tapetum layer is the layer which helps the microspores or the sporogenous tissue by providing nutrition. So जो sporogenous tissue है, जो microspores हैं, जब वो divide करते हैं, जब वो pollen grain बना रहे होते हैं, they require a lot of nutrition, lot of carbohydrates, different type of proteins, material like sporopollenin. Now all these components, they are provided by the tapetum. This is a structure when anther has dehisced. Now what happens, you can see that the wall layers, they have ruptured and center में जो sporogenous tissue था, अब वो pollen grains बन गया है. They have changed. So this is a later stage का photograph. Now again, this slide is just summing up what I told in the previous slide. When you read this, just draw the structure and put it in the chronological form. अच्छा, पहले ये हुआ, फिर develop हुआ, then the sporogenous tissue matured, and when sporogenous tissue matured and the pollen grains were formed, then the wall layer started to wither off and dehiscence भी साथ साथ होने लगा. The tapetum जो layer होता है, the cytoplasm of the tapetum is dense. When I say dense, that means it has a lot of cell organelles. And it is a very active layer because it has to constantly give proteins, carbohydrates, sporopollenin into the anther locule. And the tapetal cells are often polynucleate. That means they have more than one nucleus. What happens inside the sporogenous tissue? Tapetum cells में जो wall layers है, उसमें क्या होता है, ये हमने अभी सुना. Now let us understand, जो center में core sporogenous tissue है. The sporogenous tissue is the tissue which is actually going to form the pollen grain. These cells, they are larger in size as compared to the other cells. They become isodiametric and they undergo meiosis. Now students, you should pay attention in this process. In the sporogenous tissue, the process of meiosis happens. In meiosis, the chromosome number that each daughter cell gets becomes half. So when we have 4 microspore mother cell or the pollen microspores or pollen mother cell, so these have half the number of chromosomes as compared to what was in the sporogenous tissue. Now the 4 microspore which are formed after meiosis, they remain together, and this 4 together, the structure is known as a tetrad. Tetra means 4. So the microspore are 4 in number, together they exist as a tetrad, and each microspore is haploid. फिर as the anther matures, the pollen grain, the microspores, they separate from the tetrad और वो अलग अलग हो जाते हैं. And when they separate from the tetrad and they lie in the anther locule, then they become the male gametes or the male gametophyte. And these pollen grains, they further differentiate to form a functional male gametophyte, and they go to the stigma, land on the stigma, and then they carry out the process of fertilization. Now there is a large variability in the type and arrangement, the wall size of the pollen grain. The pollen grain, a usual pollen grain, it exists, the usual size, it ranges between 20 to 50 micrometer, and you can see on your screens, these are some scanning electron micrograph of pollen grains of different plant species. So the wall is very different, the size is different, so you can see that there is a lot of variability.
Yes.
This variation is majorly due to what happens in the pollen wall. Now the pollen wall is a very, very interesting structure, and it is also functionally very, very important. The pollen wall, it helps in the germination of the pollen grain, and also it helps the pollen grain to go and land on the receptive stigma, which is the female sex organ of a flowering plant. Now the two layers of the pollen grain, they can be defined as exine. Exine is towards the exterior. So exine, exterior. जो बाहर वाली लेयर होती है pollen grain की, उसको exine बोलते हैं, और एक अंदर उसके लेयर होती है, उसको intine बोलते हैं. तो inside intine, outside exine. You can learn it like this. Now the exine is a very ornamented kind of a structure. उसके या तो वो spikes होते हैं, या उसके अंदर convoluted होती है, up down होता है, it is porous and it is colored, because that is why pollen grains, they usually appear yellow, orangish, reddish in color. So the exine, it has sporopollenin. Sporopollenin is a very interesting chemical. It is a very sturdy chemical. बहुत resistant chemical होता है sporopollenin. और pollen grain की coating होती है sporopollenin से, जो कि exine के ऊपर मिलता है. And what happens is, because it is resistant, so the pollen grain also is resistant to degradation by acid, alkali or enzymes. I would like to tell an interesting concept to the student, that जो हमको fossils मिलते हैं कई बार, so fossils जो हैं, they are very very old. They date back millions and millions of years old. And you would be surprised, Rahul, because of this sporopollenin, pollen grains हमें intact मिलते हैं, and those pollen grains actually help us in studying how plants have evolved, because sporopollenin has kept the pollen grains intact. So it is that sturdy a chemical. It does not allow the pollen grain to degrade, neither by acids nor by rainfall nor by naturally ऐसे रहता है sporopollenin की वजह से. ठीक है. So sporopollenin is a very interesting chemical which is present on pollen grains. Okay. And also it helps the pollen grain to maintain its viability and fertility. उसके अंदर कुछ chemical compounds होते हैं जो उस stigma पे जब land करता है, तो that time the pollen grain is able to able to germinate on the right stigma because of these chemicals. Okay. The pollen wall exine, जो outermost layer होती है, उसके ऊपर छोटे छोटे small regions हैं जिनको हम कहते हैं germ pores. Now these germ pores, on these regions जो wall है वो बहुत thin होती है, because from these germ pores the pollen tube is to be formed. So when the pollen grain germinates, the pollen tube emerges. So it emerges from the germ pore. That means the plant is very smart enough to leave certain regions, because it knows that the pollen tube has to emerge from the pollen grain. So those regions are not the regions which are totally covered up with the exine. This was about the exine. Inside the exine there is a thin layer which is known as intine. It is also known as the inner wall of the pollen grain, and it is continuous and made up of cellulose and pectin. So this is a very simple layer, intine. Now after wall, after we have discussed what happens and what is the function and structure of pollen wall, let us move inside the pollen cytoplasm and the pollen nucleus. When after the process of meiosis microspores are formed or pollen mother cells are formed, those pollen mother cells, they further undergo division. उस nucleus में भी एक mitosis division होता है पहले, and you can see on your screen the first image, the topmost is the microspore mother cell or the pollen mother cell. The pollen mother cell, it enlarges in size, vacuoles are formed और nucleus थोड़ा सा prominent हो जाता है. Third image आप देखेंगे in the sequence on your right, then there are two cells eventually they start to develop. First जो nucleus होता है वो shape उसका change हो जाता है. The red asymmetric spindle kind of a nucleus is formed. That it changes its shape and then it divides. Division is very peculiar and two cells are formed. The larger cell is known as the vegetative cell and the smaller cell is known as the generative cell. Now this stage is known as the two-celled stage of a pollen grain.
Okay.
The generative cell and the vegetative cell both have their separate cell wall and membrane. You can see it in the figure, it is very clear. The vegetative cell is larger, it has more food material, reserve food in the form of starch,
Okay.
and the nucleus becomes irregular after maturity. The generative cell, it is small, it lies or floats in the vegetative cytoplasm. So it is actually a component of the vegetative cell. cytoplasm के अंदर generative cell एक अलग से cell बन के रहता है, वो spindle shape होता है, and the generative cell cytoplasm and nucleus also, the nucleus is prominent and the cytoplasm is dense. 60% of flowering plants, they shed their pollen grain, मतलब anther जब dehisce हो जाता है तो 60% of angiosperms, the pollen grain are in the two-cell stage. This is the stage when majority of the pollen grains are shed. In the rest of the angiosperm, the generative cell है, that will again divide mitotically to form two more cells, and those two cells are known as the sperm cell 1 and sperm cell 2. So those two are the male gamete, and when the pollen grain is shed in the 3-cell stage, then the pollen, those are known as the 3-cell stage pollen grains. Now this was about the development of the male gametes in angiosperm. What happens in the wall and what happens in the inside the pollen grain. These two processes, अंदर का cell division and formation of the pollen wall, take place simultaneously, so that when the pollen grain is shed after the anther dehiscence, so it is functionally a complete unit. Inside also it is competent to bring about the process of fertilization, and outside में जो उसकी exine है, it helps in pollination and also pollen germination. So this is about the development of the male gametophyte or the pollen grains. Pollen grains are very, very important for plants. We all know very well why. But they also have a lot of other functions or factors to their existence. Now, pollen grains of many species, nowadays it is a very important and an upcoming source of supplement. It provides a lot, it has a lot of nutritive value. So nowadays, like the supplements of vitamins and other proteins, you get supplements which are tablets or syrups of pollen grain. They are rich in nutrition. But they also have a flip side. Several pollen grains, they cause allergic reactions. We all develop sneezing, allergy in nose, running nose, itchy eyes when we are exposed to pollen grains. Particularly if you talk about north of India in the month of January to March and also in April, when the dry pollen grains are there in there. So that time is the one where a lot of pollen allergies are caused. So they also cause bronchial afflictions, they cause asthma, they cause bronchitis and other respiratory illnesses in children as well as in adults. Then I would like to mention that pollen grains also have a lot of ecological functions. Parthenium is a very popular and a very common invasive plant that is creating a lot of problem to the native flora of India. And when during the time of the Green Revolution and before that, we used to import wheat from Mexico. In those wheat, Parthenium pollen grains came to India. And then they kind of fertilized with the Indian varieties, and now we have a very invasive Parthenium species in India. So pollen grains are not only something which are important to the plants. But for humans also, they are good because they are source of nutrients.
Okay.
They are not so good because they cause a lot of allergic reactions in us, and also they contribute to a lot of ecological functions.
Okay.
So pollen grains are a lot to just small structures which are found on flowers.
Okay.
Now, this was about what are pollen grains, how they develop, how the process of microsporogenesis take place in angiosperms, what all structures are involved. So, starting from the differentiation of the floral primordium till the formation of a mature 2 or 3 celled pollen grain, a lot of processes are involved. And all these processes, they form a part of the pre-fertilization events when we talk about the male gamete or the male gametophyte in angiosperm. The second component or the second aspect which is equally important in the pre-fertilization event of flowering plant is the development and proper maturation of the female gametophyte or the megasporangium. Now, we are going to discuss that. But before we move on to what happens during fertilization, a very important term that we all should remember is viability. Now, pollen grains, they have to land on the stigma and germinate. But can all pollen grains germinate all the times? No, they cannot. That is why pollen viability is a very interesting feature of the pollen grain, which makes it very different from all other pollen grains, and also it is responsible for proper functioning of the pollen grain. So, viability is species specific. Some species like cereal pollen grains of wheat, maize, rice, they are viable only say for 30 minutes or so, half an hour.
Okay.
And pollen grains of certain species like Rosaceae, Solanaceae, Solanum nigrum, Vidania, they are actually responsible, they may be viable for some months even, weeks to months. So, this variability is also responsible for reproductive fitness of a species.
Okay.
Then, because pollen grains are very important for the process of fertilization, nowadays we have pollen banks, like we have seed banks.
Okay.
So, pollen grains, they can be stored at minus 196 degree
All right.
in liquid nitrogen.
Nitrogen. Okay.
And then when we have the receptive stigma, then we can conduct breeding experiments, fertilization,
Okay.
cross-fertilization experiments
Okay.
in order to get better varieties. So, there is a lot of functional relevance to pollen grains. They are not just small structures, but they are functionally extremely important structures from breeding perspective, from human health perspective, and from ecological perspective.
Okay.
Now, we move on to the female reproductive structure in an angiosperm.
Okay.
Pistil is the complete structure which is a part of the gynoecium, and gynoecium is made up of carpel. So, if a gynoecium has one carpel, it is known as monocarpellary, or it has many carpels, it is known as multicarpellary. You can see on your screen, students, we have an example of a monocarpellary gynoecium, which is papaver or poppy, and also we have Michelia, which is a apocarpus or a multicarpellary. Free carpels are there, it is multicarpellary and free carpels. So, carpels, if they are fused, the term that we use to define them, they are known as syncarpus. Syn means fused,
Okay.
and free means apo. So, you can see that the carpels on the right hand side, the Michelia species, they are all fused, they are all free from the surface, from the top,
Okay.
and they are all on one stalk, but they are not bound because the ovaries are also free.
Free.
So, that is a apocarpus structure.
Okay.
Moving on to the pistil. Now, each pistil, it has 3 major structures. One is stigma, which is the topmost region.
Okay.
Then the center long part or a tube-like structure, which is known as the style. And the lowermost structure, it is swollen, it has ovules, and it is known as the ovary.
Ovary.
The stigma is the region on which the pollen grain lands. and the pollen tube, after germination, it moves through this tube-like structure style and goes to the ovary. Inside the ovary, it releases the male gametes into the female gamete, and then the process of fertilization takes place. Now this structure on your screen, it is a typical pistil which is from hibiscus. Hibiscus or gudhel in Hindi we call it commonly. So all the students might have seen this flower. When you see this flower next time, just observe it. Just see, and you can see a very nice and an interesting stigma, a long style, and also ovary at the bottom. Thalamus is the fleshy structure on which the ovary is placed or born. Now what happens in the ovary? How is the ovary functioning and what is the structure of the ovary? The ovary has a cavity which is known as a locule, and this locule, it has many ovules which are known as microsporangia. Now these, they arise from the placenta, and each ovule is known as the megasporangia, and it is attached to the stalk or the main stalk by funicle. So these are the terms, students, which you need to remember when you are describing the megasporangium or the detailed structure of an ovary.
Okay.
Then again some more terms are there. The region from where the funicle or the tip is attached to the ovule, that is known as hilum. And each ovule, it has integument. Integument means covering. So the integuments can be 1 or it can be 2. Okay, so it can be bitegmic or unitegmic ovule. The integuments are known as outer integument, and the inner one is known as the inner integument. The region at which the integuments are absent is the region from where the pollen tube is going to enter the ovule. So that region is known as the micropyle. It is a small opening. Micropyle is the opening at the tip of the ovule from where the pollen tube enters. And the opposite structure is known as the structure at the opposite end is known as the chalaza. The integuments inside, it has nucellus. Nucellus is a structure which is nutritive in function. It has a lot of reserve food material, and this mass of cell, it actually houses the megasporangium or the embryo sac. It has the embryo sac जिसके अंदर embryo development होता है आगे जाके. Now female gametophyte और जो embryo sac है वो nucellus के अंदर है. Nucellus के बाहर इंटेग्यूमेंट्स हैं, एंड इस कंप्लीट स्ट्रक्चर को हम मेगास्पोरेंजियम कहते हैं, जो कि ओव्यूल के अंदर होता है. फाइन. नाउ दिस स्ट्रक्चर, दिस प्रोसेस ऑफ मेगास्पोरोजेनेसिस, हमने पॉलन ग्रेन के टाइम पे माइक्रोस्पोरोजेनेसिस पढ़ा था, जिसके अंदर एक स्पोरोजीनस जो टिश्यू होता है, हर एक स्पोरोजीनस सेल के अंदर मियोसिस होता है. मियोसिस से 4 माइक्रोस्पोर या पॉलन मदर सेल बनते हैं, और वो पॉलन मदर सेल थोड़ा सा चेंज होके पॉलन ग्रेन्स बनते हैं. नाउ दिस पर्टिकुलर सीक्वेंस ऑफ इमेजेस, इट इज शोइंग यू हाउ द प्रोसेस ऑफ मेगास्पोरोजेनेसिस टेक प्लेस. एक एंजियोस्पर्म में मेच्योरिटी पे, एट मेच्योरिटी, अ टिपिकल एम्ब्रियो सैक, इट हैज 8 nuclei and 7 cell. So a typical embryo sac, it is 7-celled, 8 nucleate in its formation. And सबसे पहले you can see the first image which shows parts of an ovule. Only a single megaspore mother cell is observed. Unlike microsporogenesis, मेगास्पोरोजेनेसिस एक ही मेगास्पोर मदर सेल में होता है. माइक्रोस्पोरोजेनेसिस, pollen formation happens in many cells. Sometimes there are thousands to lakhs of pollen sporogenous cells. This ovule, it divides. It divides meiotically. Again, students, remember, meiosis is happening. So the 4 nuclei which are formed they will be haploid. They will have half the number of chromosomes.
Okay.
So the megaspore dyad is formed, which have 2 cells, and the megaspore tetrad is formed. Tetrad in megasporogenesis हमेशा लीनियर होता है, मतलब all the 4 cells formed after meiosis, they are arranged in a line or a sequence. You can see the third image, 1, 2, 3, 4. These are the 4 cells of the megaspore tetrad. These 4 cells, if you look at the diagram at the lower side, left hand side, bottom, then you can see 2, 4 and 8 nucleate stage of the embryo sac. Now one of this megaspore tetrad, only one of this cell will be functional. 3 cells degenerate हो जाते हैं. Okay? मेगास्पोर टेट्राड में से 3 cells degenerate हो जाते हैं और जो एक सेल बचता है, that cell it divides mitotically now because already meiosis has happened. Haploid है.
Okay.
उसके अंदर 3 माइटोसिस होते हैं, 3 माइटोटिक इवेंट्स.
Okay.
एक माइटोसिस से 2 सेल बनते हैं. आप देख रहे हैं दोनों अलग अलग एंड्स पे हैं. नीचे दूसरे माइटोसिस से 4 सेल्स बनते हैं और तीसरे माइटोसिस के बाद 8 सेल्स बनते हैं. So 8 nucleate, 8 nuclei are there.
Okay.
And these 8 nucleate cells, these 8 nuclei, they rearrange themselves to form the functional embryo sac. अब वो rearrangement कैसे होता है? When you see the last image on the in the right hand side bottom में last image, that is a typical mature embryo sac. 3 nuclei, they form antipodal.
Okay.
At the chalazal end, जो टॉप में दिख रहा है आपको.
Right.
3 सेल्स हैं वो, 3 न्यूक्लियस हैं.
Okay.
सबसे लोअर पे माइक्रोपाइलर एंड पे जहां से पॉलन ट्यूब एंटर करती है, रिमेंबर माइक्रोपाइल इज द रीजन जहां से पॉलन ट्यूब एंटर करेगी along with the male gametes. That point in that region there are 3 cells. Those 3 cells are known as the egg apparatus. The egg cell is only one. Remember students, only one functional egg cell is there and 2 cells are there which are known as synergids. These synergid cells, they have small tubule like structures which are known as the filiform apparatus. So, pollen tube enter करती है synergid में and the egg cell is there behind which brings about the fertilization with the male gamete. 3 cells at the micropylar end, 3 cells at the chalazal end. 2 nuclei are left. These 2 nuclei together exist as the central cell. ये दोनों nuclei एक ही सेल में होते हैं, इसीलिए nuclei इसमें 8 हैं लेकिन cells इसमें 7 हैं. Because in the center the 2 polar nuclei, they are together in the same cell wall and that forms the central cell. So what we have a 7-celled 8 nucleate embryo sac in which you have the embryo, the egg apparatus towards the micropylar end and the antipodals at the chalazal end. Again, this is right up for the same image.
Okay.
That a typical female gametophyte, it has 8 nuclei and 7 cells. 3 cells at the micropylar region, they form the egg apparatus. And it has egg cell or 2 synergids. The synergids, they have special structures at the micropylar end which are actually invaginations of their wall. These are known as filiform apparatus. इनका फंक्शन होता है कि ये pollen tube को गाइड करती हैं. The other end it has chalazal end that they are known as the antipodal cells, 3 cells and center में 2 large polar nuclei. So this is about the development and arrangement of a female gametophyte.
Okay.
In this particular session, we are going to stop here because at this point, the pre-fertilization events, they are stopped.
Completed.
They are completed.
Okay.
So after these events, the next set of events are the fertilization events.
Okay.
So what has happened in the pre-fertilization events? Just a recall that microsporogenesis in the anther and megasporogenesis in the pistil, it has happened, and it has led to the formation of the male and female gametes.
Okay. One question we have received, one of our user on YouTube saying, why is gamete is chief?
Why?
Gamete is chief.
Gamete means fusion.
Yes.
Fusion is required in order to give genetic variability.
Okay.
I will give you a very simple example. If you just propagate plants through vegetative means, you have cuttings or through leaves, even like their bryophyllum and all, leaves पे भी vegetative growth होता है, tubers से होता है, there are many mechanisms.
Okay.
Those plants, they are exactly replica of the mother plant.
Okay.
Okay. Now what if the mother plant is prone to some infection, or it is ill, or it is not nutritively competent? So that character will go on from each generation to the next generation.
Okay.
When syngamy happens, fertilization and fusion, it ensures that the best of characters from the male and the female parent are going into the new progeny.
Okay.
So the chances of survival, the vigor, the growth of the progeny is always better when plants reproduce sexually, because species, they have to live for millions and millions of years.
Right.
So if a disease or if an incompetence is set in one generation, and if we have only vegetative reproduction, so the chances of the species survival, they diminish over the years.
Right. So I hope our user, we don't know the name of the user because they have some different username.
Yes.
So I'm sure you have your answer now. और अब यदि कोई और भी क्वेश्चन रह गया हो प्री फर्टिलाइजेशन से मिलता जुलता, जरूर उसे ईमेल करिएगा. क्लास 12 का ईमेल आईडी आपके पास है. अब नेक्स्ट जो सेशन होगा इसको अच्छे से पढ़िएगा, कंसेप्ट क्लियर करिएगा, सो दैट वी कैन अंडरस्टैंड फर्टिलाइजेशन इन अ वेरी वेल वे, और एक बेटर आपके पास अप्रोच होगी उस पूरे पर्टिकुलर कॉन्सेप्ट को समझने के लिए, ताकि आपका जो पूरा एक क्रोनोलॉजी है वो बनी रहे और आपके जो मार्क्स हैं वो कंसेप्चुअलाइज्ड तरीके से बेहतर आएंगे.
योर अंडरस्टैंडिंग अगर बेटर है तो इट इज वेरी इजी. मार्क्स आर सेकेंडरी.
यस, हमेशा याद रखना है आपको कि मार्क्स सेकेंडरी रखिए, अपना कंसेप्चुअलाइजेशन जो है वो क्लियर रखिए, उसको बेहतर बनाइए. डॉक्टर पूजा, आप यहाँ तक आई, आपने आज का ये प्री फर्टिलाइजेशन का सेशन पढ़ाया फ्लावरिंग प्लांट्स के लिए. थैंक यू वेरी मच फॉर.
थैंक यू राहुल, इट्स अ प्लेजर.
थैंक यू वेरी मच, आपने अपना टाइम दिया. हमारा ये मैथमेटिक्स, हमारा ये बायोलॉजी का जो सेशन है यही समाप्त होता है. आज के हमारे सेशन यही समाप्त हो रहे हैं. आप हमारे साथ यूं ही जुड़े रहिए और हमारे तमाम जो प्रोग्राम्स हम आपके लिए लेकर आते हैं, लाइव सेशंस लेकर आते हैं, उसके लिए हमारा जो सोशल मीडिया हैंडल्स हैं उन्हें ज़रूर फॉलो करें. ट्विटर पर, इंस्टाग्राम पर और फेसबुक पर भी आपको समय समय पर नियमित तौर पर स्केड्यूल्स की जानकारी मिलती रहेगी. फिलहाल मुझे और मेरी पूरी टीम को दीजिए इजाज़त. नमस्कार.